Chapter 17, we're going to focus primarily on eukaryotic genomes. And I just wanted to point out to begin with that there's some big differences between prokaryotic genomes and eukaryotic genomes. First and foremost, eukaryotic genomes are larger and they've got more genes. And I'll show you some numbers here in just a second. They also have a lot more regulatory sequences. Um, I think it's fair to say that regulation of gene expression in eukaryotes is more complex. Um, and this comes down to the fact that, prokaryotes have this operon system where you have multiple genes sharing single promoters and, maybe a single regulatory sequence controlling the expression of multiple genes, whereas in eukaryotes, it's more on a gene by gene by gene basis. So every gene will have its own promoter and gets regulated independently most of the time. Um, I should also point out a lot of eukaryotic genome is what's called non-coding DNA. So this just means DNA that's not coding for proteins, that's not genes. So that would include what I was just referring to, promoters and other regulatory sequences, but also repetitive DNA, which I'll talk about here in a minute. Things like introns, which are the extra sequences that you find actually within eukaryotic genes. And we've mentioned this previously, but just to remind you, prokaryotic genomes consist of just a single circular chromosome, whereas eukaryotes have multiple linear chromosomes. And each chromosome has to have, origins of replication. It's going to have its own centromere. It's going to have these repetitive telomeres at the ends of the chromosomes. So those are some very significant differences between prokaryotic and eukaryotic genomes. I mentioned eukaryotic genomes are larger, and this small table gives you a feel for increasing genome size and increasing gene number as we sort of progress uh, through the evolutionary scale from, arguably primitive single-celled prokaryotic cells like bacteria. We've got genome sizes ranging from, 500,000 bases up to, let's say, 4.5 million. Anywhere from say 500 to 4,000 genes in the bacterial world. Then we get into like single-celled eukaryotes, like some yeast. This is Baker's yeast and fission yeast. Now the genomes are up to about 12 million bases. We've got about roughly 5 to 6,000 genes. And then we've got our multicellular eukaryotes, plants and animals. And you can see the genomes now are generally much, much larger. And we've also got significantly more genes in the genome. What's kind of interesting though is if you look at the percentage of the genome that actually codes for proteins or the percentage of the genome that is genes, that percentage is very high in prokaryotes and bacteria here, right? 80 plus, really 90% of the genome is genes. Whereas once you get into the multicellular eukaryotes, it's a very small number, right? Humans, 1.2% of our genome is genes. Again, just emphasizing that fact that our genome is made of lots of other stuff, right? Repetitive sequences and regulatory sequences, etc. Now, we're going to talk a little bit about the human genome, but before we get there, I wanted to point out that a lot of, arguably, really even most of what we learned about eukaryotic genomes has not come from studying the human genome, but has come from studying the genomes of what are called model organisms. So these are organisms that are easy to work with in a lab setting, easy to perform experiments on, unlike humans, right? We can't easily do experimental biology with humans in most cases. So there's this sort of small handful of very, very well studied model organisms where not only have we sequenced and analyzed their genome very thoroughly, but we have done, tons and tons of other studies, manipulated genes and, done crosses looking at genetics and, really you name it, it's probably been done. So some of the more popular, quote unquote, popular model organisms would include things like baker's yeast, Saccharomyces cerevisiae, C. elegans, which is this microscopic nematode worm, multicellular eukaryote. Of course, the famous fruit fly, Drosophila, we talked about being used for genetic studies, but it's used in all kinds of developmental and molecular biology studies as well. And probably the most famous plant that you've never heard of is this little it's a thale crest called Arabidopsis, a little weed. But again, the whole idea with these model organisms is we can do these experiments and really understand these things inside and out. And of course, these are not economically significant organisms or medically significant organisms, but we can take what we learn here and hopefully apply those to other, let's say economically significant crop plants, or we could take something we learn in fruit flies and maybe even apply that to human biology. Um, So, just to give you another example, this is not coming specifically from what would really be considered a model organism. This is something that came out of the Dog Genome Project, but it illustrates the fact that what you learn studying animals and animal genomes, say mouse would be another good example of a model organism that wasn't included on the last slide. What we learn from studying these animal genomes may be able to be applied to human biology and medicine. So in this case, the particular point of interest that popped out of some studies of the Dog Genome Project is related to this myostatin gene, which encodes a protein that will block muscle growth. And what you find is in some of the dog breeds that have very, very well developed musculature, we find a mutation in the myostatin gene that basically inactivates the protein, so you're not getting that inhibition of muscle growth. And in very dramatic examples, you can end up with something like this, where you have a quote unquote normal whippet on the left, and here's a whippet with a mutation in its myostatin gene, right? So it's just got this incredible muscle mass development. And so we observe this studying animals, and it's a bit of an oddity. You might think, okay, well, how does this apply to human biology and medicine? Um, well, if you think of muscle wasting diseases, something like MS, for example, it may be possible to manipulate the myostatin gene or its expression based off what we observed in the dog genome and studying dog biology, and use it to help treat some of these diseases in humans. So again, the whole gist here is studying model organisms, or other plants and animals that we might not think of as economically or medically important, can actually be very, very beneficial in the long run. All right, so what have we learned looking at eukaryotic genomes? Well, if we just focus in on the genes, um, we find that they can be grouped into these, their functions can be grouped into these uh, pretty regular categories no matter what eukaryote we're looking at. So we see about a quarter of the genes in a eukaryotic genome we would consider being important for general metabolism. So all these different biochemical pathways involved with building up and breaking down carbohydrates and lipids and proteins, etc. We've got about 20% each for basically maintaining the DNA and controlling expression of genes and maintaining the membrane and transporting things in and out of cells across membranes. We have about 15% that's important for maintaining cell structure and the structure and function of organelles. Got about 10% of the genome involved with regulating the cell cycle, and then about another 10% that seems to be critical for sending signals within and between cells. And again, this is pretty consistent, no matter whether you're looking at a plant or a yeast cell or a human cell, you will see roughly the same breakdown. But I did want to remind you that a very large portion of eukaryotic genomes is not genes, right? Remember, in humans, less than 2% of your genome is actually genes. And a large portion of eukaryotic genomes has been found to be repetitive sequences. And in fact, there's so much repetitive DNA that's been categorized into these groups. So there's what's considered highly repetitive or moderately repetitive sequences. Highly repetitive, these are going to be usually very short, like less than 100 nucleotides that just get repeated back to back to back, hundreds or thousands of times. And again, there's no genes in there. We're not exactly sure what its purpose is, but we do use some of these highly repetitive sequences as the basis for DNA fingerprinting. Because whereas the sequence of genes tends to be very, very identical across most individual humans, um, the the sequence and the number of these repeats can be very distinctive and can be used as a unique identifier. And then we have what are called the moderately repetitive sequences. Um, so these will only be repeated, maybe up to 1,000 times. Still a lot of repeats. And these actually will include some genes. So we have genes that code for the transfer RNAs and the ribosomal RNAs that are so important for protein production. Because you have so many ribosomes in your cells making proteins all the time, you need many many hundreds of these genes. All right, now let's focus in on the Human Genome Project. As we said, this was a 20-year effort completed in 2005. And of course, we've learned a lot since then, in analyzing the human genome, although I think it's fair to say we still have a long way to go. We're definitely not at a complete understanding by any stretch of the imagination. But I just wanted to pull out a few interesting facts that kind of jumped out early on as we completed this sequencing project. So, as I've already mentioned, it was kind of surprising to find that such a small percentage of the human genome was genes, right? Less than 2%. And we found that humans have about 24,000 genes. That was also a little bit surprising because there's some other multicellular eukaryotes that have significantly more genes than that. But we'll talk about why this may be the case here in the next bullet point. Um, we know from studying proteins in human cells that we definitely have more than 24,000 proteins. So this means that each of these 24,000 genes has to code for multiple proteins. And the way this would happen is through that phenomenon called alternative splicing that we've discussed, right? Where, remember, every eukaryotic messenger RNA gets spliced, and by splicing in different combinations, we could actually produce different proteins from a single gene. The average gene is about 27,000 nucleotides or base pairs in length. Um, every human gene, really every eukaryotic gene, as far as I'm aware, that's been studied has introns, right? Those intervening sequences that are found in the middle of genes. Um, Over half of the human genome is either transposons, which I mentioned earlier in this chapter, or those repetitive sequences that I was just talking about. And surprisingly, we tend to focus on the differences between people, but at the genome level, we are very, very, very identical, right? 97% of our genomes is identical between people. So it's just that 3% that accounts for all the observable differences. And maybe not surprisingly, genes are not evenly distributed throughout the genome, right? Of course, you have 46 chromosomes or 23 pairs in your genome. The Y chromosome is the smallest of the chromosomes, so it has the fewest genes, about 230. Chromosome 1 is the largest chromosome. It has the largest number of genes, about 3,000. So, what are we able to do with the human genome? It's kind of amazing that we can basically read our own instruction manual, but then the question becomes, okay, well, what good is that? What can we do with that? And I think that's still a bit of an unanswered question. We're still trying to figure that out. But pretty quickly, we did find a few useful things that could be done by looking at human genomes. Um, so, pretty early on, after the human genome was completed, researchers were able to find these DNA sequences, um, essentially like unique sequences. You could consider them mutations if you wanted, that were linked to specific diseases. And these are called SNPs, which stands for small nucleotide polymorphisms, which is basically just a fancy way of saying uh, DNA sequence changes or differences. But again, the whole idea is by looking at lots of different individuals' DNA and sequences, or maybe not their full genome, but at least parts of their genome, researchers identified these unique sequences and were able to find specific sequences that were associated with specific diseases. And this is obviously useful information to have. So here's just a few examples. They found SNPs that were linked to breast cancer, heart disease, heart attack, obesity, diabetes, prostate cancer. And, if you're heterozygous, if you have one copy of one of these SNPs, it increases your risk a certain amount. If you're homozygous, it's going to increase it significantly more. But these numbers are not insignificant, right? You would want to know if genetically speaking, you had a 65% or for sure a 270% increased risk of developing diabetes, right? You might be able to take early action, right? Watch your diet, monitor your blood sugar levels more closely, and try to prevent diabetes from developing. So that's something useful that could be done. And kind of taking this a step further, a lot of this has come out of cancer studies, but researchers have found that your unique genetics can affect the way that you may respond to particular drug treatments. And this has led to this field called pharmacogenomics, which is basically trying to individually tailor drug treatments um, to specific patients. And so imagine you've got a situation like this where all of these individuals in the brown box here are patients. Let's say these are women that were diagnosed with breast cancer, right? And of course, there's different types of breast cancer depending on what genes were mutated in those in your cells. Um, cancers may behave very differently, and they may respond differently to various treatments, chemotherapy regimens, or other drugs. So if we were to, let's say, take a biopsy of the breast cancer from all these patients, right? Sequence at least some of the cancer cell DNA, we might be able to subdivide these into now two different groups. So this large group, we found, okay, based on the specific type of cancer they have and some of the genome sequences in those cancer cells, data shows they're going to respond better to treatment A. Whereas the smaller group, because of their unique cancer type, they're going to respond better to treatment B. So that would be pharmacogenomics, using that genomic information to tailor that drug treatment to be most effective. And that's a big buzzword these days. You may hear about individualized medicine. So that's really the only way we can, or not the only way, but the best way we can individualize medicine is by using genomic information to inform our choices in that regard. All right, so to end the chapter here, I just wanted to point out that because of this phenomenon of alternative splicing, where each gene is coding for multiple proteins, um, and the fact that you have what are called post-translational modifications, so after proteins are made, they get modified in different ways. Um, if you just looked at an individual's genome or an organism's genome, you're not really going to get the full picture, right? You're not seeing all the proteins that are present in the cell. And of course, it's the proteins that are really doing all the heavy lifting, all the hard work. So there's a couple of other techniques that are really important to complement genomics, and these are what are called proteomics and metabolomics. And so proteomics is an attempt to characterize all of the proteins in the cell. We would call that the proteome instead of the genome. Um, and there's various techniques to do that. I won't go into those for our purposes here. But if you understand the genome and you understand the proteome, now you're getting a much more complete picture of what's going on in an organism's cells. And then the last uh, category I want to mention is what's called metabolomics, which is studying all the metabolites in a cell, or the metabolome. And metabolites are defined as small non-protein molecules. And this is important because it's been shown through study after study that what some of these small non-protein molecules, these metabolites, are doing can drastically change the biology and the functioning of a cell. So if we really want to understand how a cell is going to behave, we would ideally know the genome, right? What's the instruction manual say? We'd look at the proteome, see what all the proteins are that are present in the cell, how are they functioning, what kind of activities are they involved in, and then how is that all being influenced by the metabolome, by all these metabolites that are found in the cell as well. And there's some very sophisticated techniques now that allow us to really try to combine these three fields into one sort of unified picture. So anyway, that is all I've got for you. I will stop it right there.